Thank you, Parker. Let's see, we're going to give a couple of minutes for people to roll in, and then we are going to start on 103. In the interim, I'm going to start welcoming all of you to today's Meet the Experts webinar series. We will continue in our re-engagement with the community. So we're hoping for a brand new 2026 with new topics in the area. We are hoping that this year we address some of the topics that were suggested by the community last year. So I think we are on time. And it has slowed down. So Parker, if you could move to the next slide. So for those of you who are joining us and are not yet familiar with the repository, or this is your first time joining the series, the repository program itself was established over 20 years ago. And our primary role is to facilitate the sharing of resources that includes both data and biospecimens that are generated from many of NIDDK-funded clinical trials and clinical studies, with the purpose to make those resources available for secondary research use to the broader community. We have lots of different resources, including imaging data, clinical data, and biospecimens. We have over 7,000 registered users, and our weekly users goes over 5,000. Our users, the ones that have been approved to access the resources, have generated over 800 products combined. Next slide. We are part of the broader NIH data ecosystem, and one of the things that we do is try to provide things that will make or facilitate the research for our secondary users. One of the things that we are hoping to have released by the later spring, is the analytics workbench that we have been working on. This is a two sort of thing. We will hope to have the cloud-based analytics environment that will be functioning with the main application, and an analytics workbench environment that will be working through the data challenge platform. This series is funded through that, or supported through the efforts that have been for data challenges, or for us to promote data science. So we are very excited of all the functionalities and all the things that we will be able to provide to our user community in the coming spring, so stay tuned. Next slide. As mentioned, this series is part of a broader data science and data science-centric movement. Our goals were to develop tools and approaches, as well as models and methods to increase data interoperability, as well as artificial intelligence and machine learning applications to augment and enhance existing data for future secondary use, and to discover innovative approaches to enhance the utility of the resources that we have for AI and ML applications. We have lots of resources available on our website, and I encourage all of you that are present today to visit our website for those that are past information, like our past challenges and future challenge opportunities, as well as past recordings from this series. Next. Not to waste more time today, we are going to introduce to you our experts for today, who are Dr. Chen Li, who is a professor in the Department of Computer Science at UC Irvine. He received his PhD in computer science from Stanford University. He also holds graduate and undergraduate degrees in computer science from Tsinghua University in China. He also received a National Science Foundation Career Award and several Test of Time publication awards. He is a part-time visiting research scientist, or was a part-time research scientist at Google, and an ACM distinguished member and an IEE fellow. He is co-founder and CTO of a startup to commercialize his research. With him today, joining him are Kung-Woo Park and Jia Dong Bei, who are PhD students in computer science, also at UC Irvine, who are working on the Texera platform, who are what they are going to be talking to us today. So without further ado, I let the experts take it over.
Yeah, thank you, Rebecca, for the introduction. I'm going to first share my screen. I hope I'm sharing the right screen. Can you see it? And let me
Yes.
do the slideshow. Can you still see it?
Yep.
Okay. Yeah, thank you for having us here. What we plan to do is I will first show a few slides to give an overview of the Texera project. And then after that, our team members, Chris and Jiadong, they're going to show a live demo. Hopefully, with the live demo, many things can become more clear. And then after demo, we are going to switch back to the deck to show a few more slides about what we are doing, other activities, and our future plan. Okay. So first, a single slide to give an overview of the Texera project. This is a project we have been working on for almost 10 years. The main vision of this project is to support data science, AI, ML as using the GUI-based workflows. And it can enable cloud services. And we know the benefits. Users don't need to install any software on their local laptop, and everything will be available smoothly through the cloud. And it supports community-based sharing of resources, including data, workflows, and later also ML models. It has many powerful functionalities, in particular, those related to collaborations like shared editing, shared execution. It has rich support of different program languages, including Python, R, Java as user-defined functions, and is always open source. And we are in the process of going through the Apache incubation phase to become, hopefully, to become a Apache officially Apache project. And the backend has a parallel engine to be able to handle a very large amount of data. So this is one slide, the overview of the Apache Texera project. I forgot to mention this work is supported by National Science Foundation, also NIH, NIDDK. So there's no surprise here. So thank you very much for your support. And this effort is part of the NIDDK-DKNet project. With this system, we are developing a computational core for the DKNet award. The main vision of enabling this computational core is to make data science AI ML tools techniques easily available to the NIDDK community through the DKNet award. And the main POs are Ashley and Xujing. I don't know if they are on this call. So now I'm going to use an example to show one of the motivations. So here's a typical case where we have a research lab, say some university, there's a PI, and in this lab we have one biologist called Alice. Alice, she does a lot of experiments in lab. Sorry, Alice is a PI, a biologist, and Alice has another bioinformatician called Sally. Sally knows both biology and also computer science, and Sally can do some coding using Python, using R to analyze sequences to compute the count matrix, and by doing a whole bunch of steps, we can produce a UMAP. And we could have another uh, bioinformatician called Bob in the same lab or possibly different lab. So, and this is a very simple use case. We don't need to go through detail. This is a one example of a sequence analysis pipeline with multiple steps, cell ranger to do counter matrix computation, and then a whole bunch of steps to quality control, normalization, dimensionality reduction before we do even more advanced analysis. And for this pipeline, Sally can just write, say, either a Python or R code, and this is a part of the code. At a high level, this program has multiple steps: data preparation, data analysis, and later visualization. Now, what are the main challenges? First of all, coding is indeed very hard, and only people who have gone through the training, they are able to write this kind of code. And then very often this kind of a code relies on different libraries of different versions, and version control of those libraries could be hard. And when you run it, you need to have a server because when data is very big, very likely your local laptop does not have enough resources, memory, CPU, or GPU to run this analysis. And then when data gets big, even if you have a server, the computation can be very slow. And plus, not everybody, not every lab can hire a bioinformatician. As a consequence, this kind of analysis cannot be done easily by many researchers. Another need in this space is really about collaboration. We want to allow people with different backgrounds to work together, biologists, bioinformaticians, and computer scientists. In our DKNet award, we do have 4 teams, and some of the teams are indeed biologists, bioinformaticians, and computer scientists or ML researchers. And the collaboration can also happen between different labs, different organizations. But very often within a lab, some senior students, they do some coding, they write some program. What they could do is they may just put the code on like a GitHub and then provide a README file, hoping that some new students can come back, can take a look at the code and follow the README instruction to repeat the experiment. We all know how hard that is. Very likely the code written by senior students will be abandoned completely. And we can see a lot of knowledge resources have been wasted because it's just scattered across different places. And it's not easy to repeat the analysis. Another motivation is this emerging trend of more powerful AI ML techniques. Are many communities like the DKNet community, people are eager to leverage the latest AI ML techniques in their research. But still, many of them may not have the necessary skills to do coding, to get familiar with how each ML model really works and how to even incorporate these kind of AI techniques into their research for those reasons I mentioned earlier. So given all these needs, also challenges, we have been developing this Texera system to support collaborative data science and AI ML using GUI-based workflows. And we have been doing the project through open source. This is our GitHub website. And what we plan to do next is to let Chris and Jiadong show a live demo to see the user experience on the platform, and then we can come back to this deck to continue the presentation. So I stop sharing, and I'll also mute my microphone. Chris is in my office. He will turn on his microphone. Yeah. Let me stop sharing.
Let me start sharing.
Yeah, go ahead.
How do I share?
Click the share button on the top right. You see it? rejoined.
Yeah, I
got it. Back. Yeah, Chris has to change his security setting. He has to rejoin the session. Sorry, Chris doesn't use Teams very often, so we often use Zoom.
Okay, sorry. Yeah, sorry for the delay. Yeah, I hope you all guys can all see my screen. So I'm going to show you a live demo of the Texera, mainly how users can use Texera to do data analysis. So the screen I'm showing you is a website called texera.dknetai-ai.org. It's publicly available to the users. So for the demo, I'm going to show you two different data analysis. So the first data analysis I'm going to show is beta cell spatial transcriptomic data analysis. So first of all, I'm going to show you what data I'm going to analyze for the demo. So if I go to the datasets tab, I already uploaded data to this platform. It's a single CSV file, which contains 7 columns. And this CSV file is a beta cell spatial transcriptomic data collected from the human pancreas. And here it has total 21 rows, and each row represents a donor, a patient who has a diabetes, and each column represents its feature. So the first four columns represent average gene expression from the beta cell, um, and the three rest of the columns are like the metadata of the patient, mainly BMI, HbA1c, and age. So for this data analysis, what I wanted to do is I want to know the relationship between this insulin gene expression with the three metadata. So what I'm going to do now is I'm going to use this data to create a workflow. So if I click the left side workflows tab, I can create a new workflow, and there are already like existing workflows that I created, but yeah, in the demo, I'll create a new one. So here now we have a empty workspace. And first of all, in the data analysis, I want to first read that CSV file that I just showed to you. So what I'm going to do is I'm going to add a new operator called CSV file scan to scan the CSV file that I showed. So if I drag and drop, then on the right side, there's a property panel to specify the property of this operator. I can select a file to scan the CSV file that I showed. So I'll click this beta cell and then I'll click the CSV file. So now it's properly set, and what I'm going to do is I'm going to first run this operator to see if it works. So in order to run it, I need to create a computing unit. The computing unit is a computing resource dedicated to the user. So whenever like user wants to run a workflow, they can create a computing resource, a specific computing resource. So I'm going to create a computing unit, and here there are like a few settings that I can like configure, like a memory size, like number of cores, also number of GPUs. But for now, like I'll keep it as a default setting and I'll create it. It will take like 10 seconds to create this computing unit. And once it's ready, then this button will become blue and activated so I can run this operator.
You can see what is the CU is. Okay, kind of see.
So right now, what's happening behind the scene is our server is all ready, but our server is running in a Kubernetes environment, and it's creating a dedicated resource for the user. So now it's ready, and I'll click the run button. And it's very quick and it's already finished. So I can open the result panel and I can click the operator to see the result. So it's exactly the same result as what I just showed in the dataset tab. So I verified that it's working. So now what I'm going to do is I'm going to add another operator right next to this operator. So as I said, for this analysis, I want to look at the relationship between insulin gene expression and the three other metadata. So I will just keep those 4 columns and remove the rest of the columns. So I'll add a new operator called projection and drag and drop. And then it's automatically connected. And on the right-hand side, I can configure this projection operator to just keep the columns that I want, that I'm interested in. So I'll just add a few, the attributes that I'm interested in. So I'll add a insulin, I'll add a BMI, I'll close it for now. I'll add a HbA1c, and finally I'll add a age column. And then I'll run it to see if it works again. So if I look at the projection operator and I open it, then now it has 4 columns. Okay, so after that, what I'm going to do next is I'm going to do a machine learning to train to get a machine learning model to predict this insulin gene expression using these 3 attributes. So first of all, what I'm going to do is I'm going to split these 21 rows into training data set and test data set. So I'm going to add a split operator right next to it and connect it. And here I can specify the split percentage. So 80% means 80% of this 21 row will become training data, and the rest 20% will become test data. So next, I'm going to add a linear regression machine learning model. So I just added this linear regression operator to train the linear regression model. And in the linear regression operator, I can specify the target attribute I want to predict, which is insulin gene expression. And I can also specify the degree of the polynomial function of the linear regression. I hope everyone are familiar with linear regression formula, but yeah, I'll specify it as 1. And now I'm going to run this workflow again. Okay, so it's ready, and it shows like the training result, mainly MAE and R2 values. And also I can see the result, which is a model name, linear regression, and also the model, the actual trained model using the data set. So yeah, that's all for my data analysis. And one of the nice features I want to show next is I can share this workflow to someone else, and that collaborator can come to this canvas and we can work together as a team. And it's a pretty similar experience as Google Docs. So what I'm going to do is I'm going to share this workflow to my colleague Jadong. So I'm going to type his email and I'm going to share this workflow to him. Also, I'll share the computing unit as well. Okay, share. Also the data set. I'm going to share the data set. Oh, okay. Yeah. So back to this workflow. Oh, what happened? I'll make it prettify. So next, Jiadong, you can join this session.
Sure. Please, can you see me on the canvas? I can
Yeah.
see you on the canvas.
Yes.
Okay.
So yeah, I can see the Jiadong's icon, mouse pointer. And I'll just keep my hands out of the laptop so he can do the analysis.
So I'd rather have a figure on top of the analysis Chris has been built. So to do that, I will go to operator panel. I will add a scatter plot to this workflow. And the thing I want to visualize is the BMI and also the insurance gene. So I will connect this visualization operator with the projection result. And in the operator, I'm going to select the x-axis as the BMI and the y-axis as the INS. And I'm all done with my editing. Now I will run the workflow. Chris, I believe you've also seen the same execution as me.
Okay, so yeah, I can see the execution and I can also see the result that Jiadong ran. Okay, yeah, okay, yeah, thank you, Jiadong. And another like nice feature is since like Jiadong modified this workflow, I can always like revert this workflow to the previous version. So I believe the previous version is this one, and maybe before adding this scatter plot, which should be. Oh. So yeah, it shows that like I can revert the version that Jiadong add an edge to before Jiadong added the edge to the scatter plot. So that's one nice feature I can show. And the next this is the end of the first analysis, and what I'm going to do is I'm going to show you the second data analysis, which is a single-cell RNA sequence analysis. So I already created this nice workflow, which is more complicated than the previous version. It's doing a single-cell RNA sequence analysis using a count matrix. So what I'm going to do is I'm going to run this workflow, and it takes 1 minute to finish. It's using a very small data set for the demo purpose. And while it's running, I can explain what each step is doing. So the first step is reading the count matrices generated from the sequence analysis, sequence alignment step using Cell Ranger in this case. And then the rest of the steps, it's creating a Seurat object. It's doing some filtering and for quality control. And then it's doing normalization, PCA dimensionality reduction, harmony integration, and clustering. So at the end of this workflow, it will visualize a nice UMAP to show the relationship between the gene and cells. Yeah, and the feature, this workflow what's different from the previous one is it supports UDF operators, user-defined function operators. Mainly, it's this whole workflow. Most of the workflow is written in R. So if I click one of the operator, for example, here it's written in R, and it's doing a filtering using some parameters and trying to keep the valid cells using the parameters. So yeah, it's already finished while explaining, so I can visualize. I'll just show you the last one. It's a UMAP. It's very scattered because it's a very small. We are using a very small data set, but it's also runnable with a large data set, and the visualization would be much nicer than this one if we use a large data set. Yeah, and yeah, that's for this workflow. And one of the important feature we want to emphasize is reproducibility. This platform, one nice feature is that I have this analysis, and if I want to, I can easily share this analysis to someone else, and that person can also easily reproduce what I did with this workflow by just clicking with just a few clicks. So I can share this workflow to the public so that any other users can view this workflow, and they can clone this workflow to their own workspace, and they can run the workflow very easily. So I'll make it public, and it's public right now. And if I go to the hub workflows tab, then I'll see that this workflow is publicized to the users. So now I'll stop sharing the screen, and I'll let Jiadong share his screen to make him to let him run the workflow. Yeah.
So I hope people can see my screen. And as Chris said, this platform support a very seamless sharing of the data and also analysis using just a few clicks. And I'm also currently on this texera.dknet-ai.org website. I can go to the hub setting with the workflows. And now I can see the workflow Chris just shared with me, or just share with the whole community. About that, and I can click to see some overview of this workflow. This is about the single cell sequence analysis after single cell sequence alignment. And also I can see the structure of this workflow from this page. And not just seeing the structure, I can even clone this work and run it by myself to reproduce the result. To do that, I can simply click this button. Now I see the whole pipeline appear on my own workspace, and I use my own computing unit. I can run the pipeline, and due to the time, I will not wait for the workflow to finish, but I hope we convey the message that on this platform, any user, they can construct a pipeline, they can easily share it with their collaborators or share it with the whole community, and other users can come to the same platform and then reproduce what other people have been done. And all this whole process is using the workflow abstraction, which is very easy to use, and also the cloud services hosting on the web browser. So I believe that's the end of our demo.
You can mute.
Sure.
Check. So thanks, Chris and Jadon, for the demo. I hope people can hear me. Let me share my screen. Yeah, and let me do the slideshow. The demo shows the basic feature of the platform. There are many other more powerful features available on the system. If you're interested, we can talk about them offline. This one shows some numbers about this project. As we said earlier, we do everything on open source community. We're in the process of going through the Apache incubation phase, and we have an active user base and developers, and we have created many workflows with many versions, many executions, and the largest deployment we had included 100 nodes. Each node has 4 cores. So in that deployment, we had 400 cores. Is parallel. We have some other use cases. This is one of the examples where we can use a platform to do a brain image analysis. Similar story, multiple people from different groups with different complementary backgrounds. They want to work together to analyze brain images, and the whole platform can facilitate the collaboration. And at the end, the final 3D model constructed from a brain can be visualized through the browser. In addition to using the system to support scientific research, we have been actively using the system in the space of education. So this is a picture we took in one classroom where we used Texera to teach AI, ML, data science skills to students not from STEM departments. Some people are from biology, from social science, math. They were able to learn those skills very quickly. Last summer, as part of the DKNet effort, we organized a DKNet summer boot camp. We did it online this time. We taught those skills to about 10 to 20 students or researchers in biology. They use the platform to learn data science. And despite the fact we have been working on it for almost 10 years, as we work with our users, we have encountered many new problems. So this slide shows some of the efforts we're currently working on. For instance, ML models are extremely important. How can we allow the platform to manage this kind of ML models? At DKNet, we have some pilot projects, and one expectation from these projects is to develop some ML models specifically for the NIDDK domain. So we would like to bring these different ML models to the platform. Then those ML models can be shared with the community. And people might be wondering, why can't we just use the platforms like a Kaggle or Hugging Face? And a few reasons. First, those platforms are very general purpose. We want to build a platform specifically for one domain, like a NIDDK domain. In addition, uh, we're doing everything through open source, and as a consequence, it's much easier for us to do more customization for each particular domain. And then another effort is to incorporate more AI ML techniques into the platform. For instance, how can we enable ChatGPT-like experience using which people can just type in some like natural language sentences, and the system can automatically use some large language model to construct this pipeline, even without even using this drag and drop operator steps. That one is ongoing. And we are making more analysis pipelines available to the community. This is a joint effort with other colleagues from Cornell, from UCLA and UC San Diego. So as we build more pipelines, we want to make these pipelines available to other researchers in the community. Then they can share, they can leverage the analysis already done by other people to benefit their research. Security, privacy are very critical. We have some ongoing effort to make the system more secure and safer, and how to allow analysis on a very large amount of data. In one of our use cases, the users are uploading like tens or hundreds of gigabytes of data. And in some cases, they can even upload 1 terabyte of data. How can we make sure the system can allow this kind of a large file upload very efficiently and then do the analysis on the platform? Another effort we're doing is many labs or campuses, their local computing resources could be limited. And in some cases, they need to elastically grab some resources on the public cloud, like AWS or GCP, or sometimes they can use some private cloud from other providers. So how can we allow the platform to dynamically grab more resources from other service providers and adopt this pay-as-you-go model? So there are a lot of research problems in that space. To summarize, the Apache Texera project is an open source system to support cloud-based collaborative data science and AI ML. And it has very strong support of collaborations and sharing. And it has a parallel engine to handle big data. And it supports multiple languages. And it has a very specific focus on, say, data science, AI, ML. So that's the last slide. So we are open to questions from the audience.
Thank you. That was a great presentation. I'm sure questions will be coming. I'm going to start with some of the questions. In NIDDK, of course, we want to make sure that this is available for the broader community. And I know that you mentioned, and thank you for mentioning that. I know that you're working towards enhanced security. If I'm understanding correctly, right now, this is available for open data, meaning that data that can be used that does not require register or controlled access. And one of the efforts we're working towards is to make this available for control access data. Is my understanding correct? Is that how we should be interpreting these efforts?
That's correct. We had some ongoing discussions with some colleagues who care a lot about say access control to be more specific. For instance, I mean the current demo we just showed, we assume the person who wants to publish data, they make the decision to just publish data. And then like say Chris has some data, he can publish data, and everybody on the community can use the data. Or Chris can share the data with another collaborator of say Jiadong, then only Jiadong can access the data. So that kind of access control is indeed already in place. But at the same time, I understand in some cases, especially in the NH domain, we need some stronger access control. For instance, some data is very sensitive. Maybe only people on the platform can do so. So that one requires some different way of securing the data access. For example, who can come to this website, right? And do we want to support this notion of groups? And then for some data sets can be only available within each group, and similar to other systems like a Linux file system, and whether how do you know the data can be indeed shared? There are many open issues, and we would like to work with the practitioners who can be very specific about the access control model, and we can do the corresponding either implementation or customization.
That's very good. We definitely would like to ensure that this process, which is, the Texera project, is available through the NIDDK-CR workbench. So definitely let us know how that progresses. Definitely, this is directed to those in attendance. If you have questions, please put them in the chat or unmute to ask the experts here in the call.
Yeah, those message questions will be in a separate window. Do we see the messages?
Yeah, we do have a Q&A, and we have here a chat as well for those that are internal to NIH. And I will moderate them. I don't see anything coming through the chat right now. Parker, do you see anything on your end that I'm missing?
I do not. But as individuals think of their questions, I'm also going to launch our end of webinar poll to get feedback on today's webinar on Texera, and then also to get feedback for future webinars as well.
Yeah, I mean, if, I can, since we're waiting for questions, I can share some websites with you. For example, the official website is texera.io. Hope that's easy to remember. This site shows information about like our education related activities. This is all the programs we have organized to do teaching, and we have a list of publications about the techniques we're using behind the scenes. So I hope you can see for that kind of user experience, a lot of technical challenges we're facing. Our team, that's the main focus of our research, because we are from the CS department. We are solving a lot of CS related techniques, and then we want to make the techniques available to users. And then we have some the source code on GitHub, right? If you're interested, you can take a look at the whole history of development. Development is very, very active. And then the DKNet website, I think Rebecca and Parker, you should be familiar with them. They are. This is really the kind of the umbrella project on top of our Texera system, right? This is also funded by NIDDK. So all these resources are available online for people who are interested.
So Dr. Li, can users go through the through the DKNet, and from there reach the Texera platform directly?
Yeah, a good question. So that's related to one of the earlier questions about security and privacy. So what we're doing here as part of this DKNet grant is we are hosting the server, right? Whatever Chris and Jiadong showed, those workflows run on a server funded by NIDDK, and the resources, the computing resources on server are limited. And that's number one. Number two, we still have some like technical issues we are solving. For example, recently our team, in particular Chris, just solved the issue of how to allow a very large binary file to be transferred more than 2 gigabytes. So long story short, there are a few technical issues we're still working on. And for these reasons, what we are currently doing is to find kind of alpha users of our platform. We invite people who are interested in the platform. You can reach out to us, and then we can invite you to the platform, and then you can use it and give us feedback. And then by working with alpha users, we can constantly improve the system as the system is more reliable, more secure, and safer, and with more computing resources. We can gradually open the site for beta users. When we try to make it completely open to the space, then you can imagine the backend computing resource should be enough. So that's a resource issue, not a technical issue. So the short answer is we are only inviting alpha users who are interested in the platform.
Oh, got you. We do have a couple questions that came on the chat. And does the platform support exporting an entire workflow code into the GitHub repository?
GitHub, let me think. Entire code into, or what the platform supports. Let me go to the website, digestive-ai.org. Right, so this is the one, Texera here. This is the one Chris and Jiadong showed. So if I log in and if I look at one workflow, this one here, I can easily download the workflow as a local JSON file. And where do you put it? It's totally up to you. So, and we also symmetrically, we also allow another user to take this JSON file and upload it back to the system. It will become a workflow. And GitHub, we didn't do anything specifically for GitHub.
So will you keep the provenance? So will you know where the data that, the provenance, do you have some sort of linkage that tells you where the data came from or is going to?
In this workflow, we depend on the meaning of a provenance or lineage. So clearly, we know this operator is reading some file from one data set. That information is available in the system. We don't keep track of where the data set comes to the platform. The user can just upload data set. And of course, the data is transferred through the whole all the edges on the workflow. That information is available to the user.
Gotcha. There's some couple other questions. I don't know if you are able to either Jiandong can see the questions.
Yeah, Jiadong, yeah. I can also see the questions.
You
because
can?
I'm looking at it.
So
I can see the
yeah,
questions.
there's a couple here. For example, the next one is there a way to share the workflows or code outside the given share or the shared project? I think that from our prior conversations, this is a no. You can only share with the people that you specify within the project or within the environment, correct?
I mean, a person A can share this workflow with person B or person C, any person. Person A can also publish the workflow on the whole platform to everybody who is available on this platform.
And that will be the public
Correct.
selection button.
Correct. I can share and I can make it public.
But that public will be only within Texera, not the World Wide Web.
Currently, it is available under the hub. It does not require people to log in. But if needed, we can change it. We can say some workflows you can see them only if you log in. That's very easy to implement. That's not a current design, but it's very easily doable based on the need of each particular deployment. That's very doable.
So if I'm understanding correctly, it's still only for within the platform, within Texera, only Texera users. will
Correct.
have
But
access to that.
yeah, but currently it does not require people to log in to see it. Like for example, if I log out, right? Suppose I sign out. I signed out, and I could look at all these workflows. They're public. It means I can see this workflow even though I have not logged in yet. But if needed, I can just add another category, which is you can see them only if you log in. We have not done that feature, but that's very easy to do if there's a need.
So Ken, I think that addressed your question there.
Yeah, I'm looking at the next question by Aaron. For data scientists who don't have prior experience with workflows used for, say, single omics analysis, do you provide resources, guides to assist those who are interested in using or exploring the use of your tool for such analysis? Good question, and that's related to another effort, which is kind of documentation. Either some like a markdown file or even some video. That one we have not done so yet. It's really a resource issue. Our time is all limited, but it's very much needed. If we had the more manpower, we would love to develop some like a simple tutorial, some documentation for every workflow. That's one. Second, as I said earlier, one of our ongoing efforts is to really introduce like a chatbot tool, and we want to use a tool to provide this kind of help. Then, if the backend large language model is very powerful, we hope that tool can provide this kind of assistance to users who don't have this kind of experience. So it's more about improving the usability of further improving the usability of the platform. Thank you, Aaron. Other questions we didn't answer?
I don't see any other questions coming through the chat or the Q&A. So with that, then I will thank you for your attendance. And Parker, if you can put the slide for the next announcements.
Yes.
I want to take again a thank you for today's experts for the wonderful presentation. I think Texera is definitely a very nice project, and we definitely want to continue the conversation so that we can offer that tool within NIDDK for control access data resources. Definitely more conversation has to be had for control access data resources and the security features. For next time, we will continue our seminar series. Next time in February, we will be speaking about practical applications. One of our speakers will be for neural networks for clinical decision-making and diagnosis. This will feature perhaps the use of AI, ML, and imaging. So please join us in February. And with that, I give you back your time. Thank you for your attendance and your attention as well. Thank you.
Yeah. Thank you for having us.